he is collaborating with the Hub University with Professor Mrinal Kanti Bhattacharya. And his title of the project is Detection and Molecular Characterization of Plasmodium and Other Hemosporidian Parasites in Bats and their ectoparasites in Telangana state, a One Health approach. I invite Professor Srinivasan to continue with the talk.
Thank you very much, madam. Yeah, as you have rightly mentioned, this is task 25 of the work package 4 of epidemiological studies and sub-package 4.3, vector dynamics and hotspot mapping for dengue, malaria, and chikungunya. And I am working along with Professor Mrinal Kanti Bhattacharya, who is an expert on Plasmodium and Hemosporidium parasites. And then when we spoke with each other regarding this particular collaborative research work, with my expertise on bats and his expertise on Hemosporidium parasites and all, we decided that we'll get into understanding how bats can be, studied or understood with respect to their potential of being vectors, because we have been aware of the fact that the bats are vectors for many zoonoses, and especially with respect to viral zoonotic challenges. So we thought that why not put our expertise together and start a project to look into things that so far very few studies, sporadic studies, have been conducted in India, in South Asia, and we wanted to start something on this particular aspect. So, am I audible?
Yes,
sir. Yes, sir.
Yeah. Okay. Thank you very much for confirming. So, as I was just mentioning that there are very few studies that have been conducted in South Asia, but globally, if we are looking at, say, for example, in the Amazon basin, in the Amazon rainforest basin, or in Southeast Asia, or in southern China, such kind of studies have been given a lot of weightage, and many studies are available. But in South Asia as such, we do not have much of studies on bats and their ectoparasites vis-a-vis the Hemosporidian parasites that they may harbor. So there was one study that we have come across, which was published in 1945, that mentioned about Plasmodium infection in bats from Northeast India. And then after a long gap, we started seeing that Plasmodium had been reported from a couple of bats in India in 1984. So we know that there is a lot of works that are being done in this particular field outside South Asia or outside India, but within India, we have wonderful diversity of bats, but nobody's paying attention towards this particular aspect, and that's what we have looked at, the data gap, knowledge gap, and then research gap, and then we started to think that let's start a project with these following objectives. We are wanting to screen bats and their ectoparasites across Telangana for the Hemosporidian parasites using both the traditional microscopic methods as well as molecular methods, and to investigate bats as reservoirs for drug-resistant malarial strain and other emerging Hemosporidian parasites via genetic sequencing. This is where the expertise of Professor M.K. Bhattacharya will come in, and to document geographic distribution of infected bats, ectoparasites using GIS mapping from field surveys, and to compile and contribute findings into the global bat Hemosporidian databases and propose target public health interventions. So we plan to conduct studies in this 5 years, collecting bats from various different locations of Telangana. I will tell about the challenges later, but we know if at all we can get known routes or areas where we can be given permission to collect the bats and all those things, we would be collecting bats from such locations. And then we would be collecting blood samples via venipuncture. We would also be screening for the ectoparasites, and if we find ectoparasites on bats, we collect them for further identification and screening. And also, those ectoparasites would be used for, screening the Hemosporidian parasites. So, whatever blood we would collect, that blood is going to help us in two ways. One is to genetically detect the diversity of Hemosporidian parasites in the blood, as well as then also microscopically detect the Hemosporidian parasites. So we are going to implement both molecular technique as well as the classical microscopic technique to detect the Hemosporidian parasites. And then Dr. Mrinal is going to look into sequencing the 5 drug resistance alleles from the plasmodiums to understand whether we have drug resistant reservoirs among the Telangana bat population. And so far, we have kind of looked at whatever we know about the bat records in Telangana based on our research, as well as some kind of publications and all such kind of things. And we have started to create the bat distribution map. And then we have approached the forest department to get the collection permit because these are not strictly a kind of protected species. Earlier, until 2024-25, we were allowed to catch bats without any kind of forest department permission, except if they occur in the forest, we had to take the permission. But now that it has been under the protection list, so we are awaiting the forest department permission to come in, and after that we'll be starting to collect the bats fresh. But in the meantime, while we are waiting for the forest department permission, we are also doing the review of literature for the bat ectoparasites and endoparasites and the hemosporidians from the other countries. And bat ectoparasites, we have some knowledge, and then we are doing some review of literature and all such kind of things. So that's about the status of work that we have done so far. But interestingly, I just wanted to mention that this particular species or this particular group of mammals are very unique for a reason that they contribute to almost about 26% of the known biological diversity of the mammals in any given area. And in India, we have as many as 137 species of bats, which are mostly divided into two different groups. We call them as fruit bats and insectivorous bats. Fruit bats, as the name suggests, are basically vegetarian, and they will be feeding on fruits, nectar, pollen, and they have good eyesight, and they have long dog-like muzzle, etc. Whereas microbats eat insects and small animals, and they use echolocation for their detection of prey or for navigation, etc. And they are unique by their physiology, by their behavior, and they are the only known mammals that are capable of flight, sustained flights, and all such kind of things. So very unique group of mammals. Not only that, but their physiology is so good and so well adapted that they are able to resist lot of organisms, microorganisms that are detrimental to other mammals and other birds, etc. Their immune systems are wonderfully well adapted to overcome any kind of infection. That is basically because of their basal metabolic rate being very, very high, and their body temperatures are within, not the core body temperature is higher. They are found everywhere in India, in Telangana, in many places we have found them. Of course, during the course of the presentation, you will come to know about them. But many a times we are not able to see them because they are nocturnal, but they are everywhere. Um, some of the bats live solitarily. Some of the bats live in groups of small troop size, and some live in colonies of very, very large numbers. And they have very wide variety of feeding behavior. Their uh, their their diet includes nectar, pollen, fruits, and the frugivorous bats or the mega bats or fruit-eating bats, as we know them, they help in forest regeneration by discarding seeds. Not only that, they also help in pollination. Whereas the microbats, which are insect-eating bats, they are known to be helping us in controlling insects that are detrimental to our health as well as to the agriculture crop. Commercial crops as well as agriculture crops have got benefited in areas where we have bat populations doing well. They are unique for a reason that unlike rodents or any other group of mammals, they have a limited, reproductive capability. Every season, they give birth to only one pup or maybe two pups. So their reproductive capability is not very high as other organisms, and they are slow breeders, and that's the reason why it becomes very, very important for us to understand the threats that are impacting them and also see that their populations are not being destroyed because recruitment of new population into the ecosystem takes very long time. As I was just mentioning that they are very useful, ecologically very, very important, and that's the reason why my team and I, we have been focusing on understanding them and trying to look at what exactly they are doing, what diversity they have, and all such kind of things. And this is for the first time. Of course, this is the second project that I am looking at with respect to understanding bats vis-a-vis human health, etc. And the threats are omnipresent. We see that there is a habitat loss that is impacting these organisms. Also, pest control activities in our agro ecosystems and commercial crop ecosystems are impacting them. We also have seen that in the name of development, many roosting trees have been felled, snatching away their traditional roosting areas, etc. And in the name of tourism, many caves, many old buildings, forts, etc., have been renovated, thus snatching away the traditional roosting places for this particular group of animals. And there is some kind of local hunting that is happening for local sustenance and medicinal beliefs. When we are looking at the bat diversity in Telangana, we have documented as many as 21 species of bats, and this particular map shows the distribution of bats in Telangana as well as in South Asia, because our research spans beyond Telangana, and we document and we do research in South Asian region. As I told you, there are as many as 21 species, and this is the distribution. They are distributed in these many different families. Vespertilionidae are also called as evening bats. All of them are nocturnal, but these are specially called as evening bats because they become active during the sunset time. So we have the most species diversity in this particular family, and then we have Epositrids and Pteropodids. Pteropodids are the fruit-eating bats. We have 3 species of fruit-eating bats that occur in Telangana. So very quickly, I'll just show you some beautiful bats that occur in Telangana. This is a greater short-nosed fruit bat. I'm not going to give you how we identify all those things, but I just wanted to create some kind of awareness amongst our colleagues about why we study bats. Because they are misunderstood, nobody is interested in studying them and all such kind of things, but also because they are very, very beautiful as well as they are very ecologically important. Uh, Theopus medius, this is perhaps one of the biggest bats that all of you might have seen during the evenings and during the night. They become active, and the maps just show that from where we have got some in the past some sample collections that we have done. And this is Rositas leschnolti, which is Leschnol's fruit bat. This is the third species of fruit bat. In fact, you would be surprised that all the three species of fruit bats occur in Osmania University campus. We have very few, specimens that we have picked up and collected from throughout Telangana, but we are aware that there are some more locations where this bat occurs. This is Rhinolophus lepidus, one of the most rarest bats in Telangana. I have collected only one specimen from Akka Mahadevi Bilam cave, and we are expecting that this is a forest species, and our forests are not that suitable for this particular species to occur. So we are hoping that future surveys are going to show us some more locations from where we might find this particular bat. Then the next group of bats, which are called as leaf-nosed bats, there are as many as 4 species of bats that we have documented from Telangana. This is the fulvous leaf-nosed bat. This is the Dobson's leaf-nosed bat. Again, we know only from one location, from the Akka Mahadevi Bilam cave, and there was another location from Bhadrachalam area, but that particular area has now presently gone into Andhra site. Then we have Hippocitrus laccadiva. Hippocitrus laccadiva is a wonderful bat. We have also picked up Hippocitrus laccadiva calls from Osmania University campus, but we have not seen any specimens or any colonies as such. But we have a colony of Hippocitrus laccadiva in Golconda Fort. Then this is Hippocitrus spuris, and this spuris is this particular bat is called as Sneeder's leaf-nosed bat, and this bat also occurs in Osmania University campus, and it is kind of a bat that tolerates little bit of human disturbance, but occurs in areas where we have good vegetation. Lyroderma lyra is a greater Indian fall swapper bat. Once again, Osmania University campus has a colony, and we are hoping that this colony is going to not get disturbed because of our developmental activities and all such kind of things. Perhaps this is the only bat that is carnivorous in nature. This is the bat that feeds on other bats, small mammals, frogs, and rodents, etc. Then we have Rhinophoma hardwicki, which is called as lesser mouse-tail bat. This is again very light tolerant bat. In caves or in old dilapidated houses or forts or wells, we have seen these bats. Again, it also occurs in our campus. Now, this is a bearded tomb bat. Bearded tomb bat has also been sighted here in the campus in Osmania University Arts College. We have a small colony of bearded tomb bat, but we have good colony of bearded tomb bat in Hyderabad, both in Kuli Qutub Shah tombs as well as our Golconda Fort. Nudiventris is a naked tomb bat. We have seen this bat from Golconda Fort. So we have a good colony of this particular bat from Golconda. And we have long-fingered tomb bat, once again from Golconda Fort as well as from Khammam Fort. We have sighted this particular bat. Then this is again a free-tailed bat or wrinkle-lipped bat. It is called as Egyptian free-tailed bat, and this bat occurs in very, very wonderful wooded area and also has been reported from city ecosystems. If you can see that, there is one locality from the Mananur area, and then there are some localities within Hyderabad from Golconda as well as from Osmania University campus. We have collected this particular species. Then we come to the largest group of bats that are called as evening bats. This is Greater Asiatic Yellow House bat, which is which prefers kind of semi-disturbed areas as well as forested tracts. We have Scotophilus cooley, which is lesser Asiatic yellow house bat, which is also occurring in Osmania University campus. Then we have Eleonoctula, Indian pipistrelle, which is one of the rarest kind of bats. These bats we have seen prefer peri-urban and rural kind of ecosystems, and we have picked up its calls from many different locations, but we were not successful in picking up samples from many different locations. So that's the reason why you see that it comes up from only one location. Eleonectulus abramus, which is Japanese pipistrelle, is again very, very rare bat. We have just collected one specimen of this from Hyderabad, northern Hyderabad region. And after that, we have never come across this particular species. Least pipistrelle is most common. Osmania University campus also has this particular bat, and this is a most common species of bats, and we have collected this bat from many different locations. So is the case with Kellard's pipistrelle. This is again very, very common bat. If you go out and you see a small bats fluttering around during dusk, this is the bat that you are seeing. And then again, we have very beautiful bat, but very rarely sighted. And both the times I have collected dead specimens, I have not seen a live bat. Both of them comes from the tiger reserves. One is from Amrabad Tiger Reserve, the other one is from Kawal Tiger Reserve. So these are all the species diversity that we have in Telangana, about 21 species. Now, interestingly, many of us do not have kind of positive imaging about bats because of they being naturally good reservoirs of many zoonotic vectors. But not only that, but also many kind of misinformation that is attached to bats. In India, the biggest challenge is there is a superstition that bats are not good. So that is something that we are working on, creating positive awareness about why bats and why we should conserve bats, despite the fact that they are good reservoirs for many organisms that can be detrimental to human health. But during the COVID and post-COVID, we have many negative imaging as well as negative perception about the bats. And what we have found out that, many a times the suspect or suspicion that went to bats later was confirmed to be something else. So it is not the bats that are creating the problem. What is creating the problem is the intermediary host that comes after the bat. So in these cases of be it SARS-CoV, the first epidemic, or MERS, or SARS-CoV COVID-19 pandemic, we have suspected the bat, but later we found that the virus has jumped from bats to some other organisms and then entered into human population. So that is something that we have to understand with respect to why we should study bats, why we should be observing bats, and all such kind of things. So this particular slide is just to make us aware that there is 98.6% of similarity between the bat, beta coronavirus as well as the human coronavirus. That doesn't mean that they are both the same. So there is a lot of kind of confusion with respect to they being the source from where the organisms or the virus has come into human beings. But as you see in this particular phylogenetic analysis, we see that there are many different organisms that are also carrying the coronavirus in them, and there are many different organisms that could be threat to spread the virus into humanity. This particular slide is just to make us aware that there are many reservoirs on whom we have to start working on whom we have to start understanding, and all such kind of things. And I just use this particular slide to make drive in a point that bats are right in the middle. They are not that much detrimental to humanity as they are thought to be. We have to pay attention towards our piggery or our sweet, diversity, or our rodent diversity, from where we have a lot of potential of many zoonosis coming into humanity. But also, we also can see on the lower end, we see that we have rabbits, we have birds, we have horses, we have carnivores. All animals are reservoirs of these virus and many pathogens. But we have to start thinking, no, have good information, correct information, and start working on all these things. These are all potential. There are a lot of people who are working on this, but very few people in India, or very few teams in India, are working on understanding the reservoir potential of all these organisms. So that brings to the end of my project presentation. And what we are trying to do is we are bringing in One Health approach and trying to understand how bats are they really detrimental in having the plasmodium and other hemosporidian parasites, and would they be spreading these parasites to human beings through mosquitoes that occur commonly in these areas. So that's the very idea of this particular project. Thank you very much for listening to me.
Thank you, Professor Chalmala. Now it is time to listen to Professor B. Ram Shepherd. His title of the project is Epidemic Spread Dynamics and Viral Containment, Leveraging One Health Surveillance and Herd Immunity Achievement. So I invite Professor Ram Shepherd to deliver his talk.
Thank you, madam. Good afternoon, everyone. So myself, Dr. Ram Shepherd. I'm from Department of Sociology, and the assigned task number is 39. The title is Epidemic Spreading Dynamics and Viral Containment, Leveraging One Health Surveillance to Achieve Herd Immunity. So this comes under One Health approach as the major project outlines and objectives meets. And hello.
Sir, please continue.
Yeah, regarding the objectives of the project, the first one is to understand the epidemic spreading dynamics. The second one is to examine the viral transmission mechanisms. Third one is to grasp the herd immunity concepts, both from modern science point of view and the indigenous viewpoint of view. And the fourth one is recognizing the importance of One Health surveillance and identifying the strategies for viral containment, and also developing the future preparedness. So these are the major objectives of the project. And understanding the pandemics, we know pandemics and epidemics, endemics and outbreaks. So there is slight difference between these four phenomena. Pandemic that is confined to one specific area. The classic example is Ebola virus in the Western Africa. Epidemic, sorry. The pandemic is a global spread of the disease, and which can spread across the globe, and COVID-19 is the classic example for that. Endemics, those are the repeated, the recurring phenomena of the disease in a specific area. Malaria, dengue, these are the endemics we can consider. And outbreaks means those are the sudden eruptions of some sort of viral contamination. So all of a sudden they come into limelight. And the reason for the these epidemics or pandemic eruption, the first one is the population density, because most of the crowded areas, those are very much prone to increase the disease transmission risk. Therefore, population may be one of the reasons. Second one is naturally population moves one place to another place to meet their own everyday work. So therefore, mobility, if we stay at one place, there is a possibility of reduction of the contamination. But mobility is invariably happens. Therefore, mobility is one of the second reason. And the third one is most of the third world countries they are having low level of immunity. So therefore, the low level of immunity naturally having the higher susceptibility. So that is one more reason. And fourth one is this environmental conditions, especially climate condition and genetic conditions or different kind of weather conditions. Because of them, this in pandemics and epidemics are spreading very rapidly, unprecedented. And coming to the transmission modes, certain diseases are airborne, certain, they come through droplets, and certain are because of the result of a physical contact, and some are vector-borne and some are food-borne. So, however, okay, most of the diseases having mixed kind of, transmission modes. For example, take the COVID. COVID happened both the droplets and physical contact, but certain diseases are coming through the food contamination, or some are directly they come through air. So the chicken facts and some other facts are very good example for such kind of academic pandemic spreadings. But there is measurement to know how the pandemics or epidemics are serious. If R naught is lesser than 1, so we can say that the pandemic is going down. If it is 1 and the pandemic, the transition is very stable, that is constantly going on. And if it is less greater than 1, naturally the pandemic is giving the alarming conditions. And there are models, mathematical models to evaluate the epidemic spread. So that particular model, many books are citing that that is SIR model. S stands for the susceptibility. Naturally, most of the population is susceptible for epidemics or pandemics, and some are going to be infected, and some are already recovering. That means they infected, but gradually they are going to be recovered by the medical intervention or natural process. But however, um, both in a manual methods, and nowadays the computers also using computer softwares also assessing these kind of models, and these models are very popular in the area of the epidemiology and public health. And applications. So if you look at the application, why we have to use this specific model means first to predict the outbreak peaks, whether the outbreak is at the peaks or not. And if it is peaks, the interventions must be very serious and stringent, and we have to pay a lot of attention. And the second one is estimate the healthcare system burdens. So countries like India are having lesser, healthcare facilities. If diseases in the peaks, how we are going to be mitigate or control, and what kind of action we are going to be take to prevent such kind of diseases. And the next one is the with the model, with the help of the models, we are going to be plan the vaccination strategies. How much vaccines we have to produce and whom we have to give the priority, whether children are aged or the working human resources given the priority. Like we are going to be get the strategies from this model. And final one is the overall medical health care or public health interventions also going to be assessed with the help of these models. And coming to the viral mutation and evolution, so naturally viruses are not constant. So they move one place to another place, and they, their mutation is unpredictable. So all of a sudden they may transmit to one farm to another farm, and sometimes we may give some medicine, but medicine may not be workable. So therefore, those are going to be drug resistant. And sometimes immune, naturally we have good immune system, but because of the mutation, our immune system may not be coping with such kind of mutations. And final one, we can say reducing the effectiveness. Though we give virus vaccination, vaccine may not be that effective. The efficacy is more or less the best example are of best examples are Delta virus during the COVID. We have seen that has a higher transmissibility and higher severity, and Omicron that has that was rapidly spread like anything, and that is more immune escapable, that has more immune escapable properties. So, however, virus is unpredictable, and because of its mutation, that's what we have to pay more attention to look over these mutations. To have the proper address or proper solution. So most of the public health schools, they are talking about the herd immunity concept, and I studied this particular concept both from modern medical point of view and the indigenous, especially Indian context point of view. Naturally, herd immunity, it comes from the animal or veterinary sciences, but surprisingly, we are applying to the human public health. So that is the uniqueness with this concept. But however, we can say herd immunity occurs when enough population become immune and they are not susceptible for the disease, and they are prone to, not going to be get any attack by that, and they are measuring, public health schools are measuring this particular head immunity through the threshold formula, that is H is equal to 1 minus 1 by RO. Here H stands for herd immunity threshold, R stands for basic reproductive number. For example, take if R naught is equivalent to 5, then we need to take more precautions to achieve the herd immunity. But however, herd immunity comes through two processes. So one is through vaccine, and the another one is natural infection recovery. Uh, of course, I have a different view towards the natural infection recovery, because unless until we give the inoculation or virulation or vaccination, and people are not going to be get the immunity. So therefore, vaccine is invariably we have to take for any kind of these pandemics or epidemics. And why herd immunity is most important? Because it protects the population, especially the people who are vulnerable and more susceptible, like people like elderly. Some people, they have some sort of pre existing diseases. Those are uh, immunocompromised people and infants. So to protect these kind of people, so definitely, so we have to think about the herd immunity, and it has its own challenges, as I stated. So a lot of people, they have vaccine hesitancy because of cultural beliefs and religious beliefs and misinformation and mistrust. Because of these kind of notions, and people, they won't take the vaccine properly. So even in the developed countries, many people who are very rigid, orthodoxical, they did not take vaccine during the COVID time, we have seen. Second, there are people who are welcoming the vaccine, but unfortunately their income, their conditions are not accepting them to, allowing them to have the vaccines. So especially low income countries, they don't have that much of economic power to afford the modern vaccines. So that's what they have their own problem to achieve the herd immunity. And the viral mutation, naturally virus every week it may change its own course of action. Then we may develop one vaccine, but it may not be workable for the next time. So that's what herd immunity has also challenged. The fourth one is warning immunity. This especially one person may take a vaccine, but in due course of time, the the vaccine developed immunity may not be that effective or that good to combat with the new kind of transmissions. So that is also another drawback to the herd immunity. And the next one is population mobility. So some people, they move across the globe within a short span of time. And if they infected with some kind of virus here, and that virus may go to some other country or some other place. So population mobility may cause some kind of drawback to the achieving the herd immunity. Last one is the weaker health systems. So naturally, we have meager healthcare infrastructure in the third world countries. So we are unable to tackle the when the eruption is in the peaks. So during that time, achieving herd immunity is one of the challenge. And we can say, but however, so far we have different strategies. So one strategy, as per literature, we can see that is non-pharmaceutical measures. This comes from the social or public health point of view. So naturally, this is using the masks, maintaining the disease distance or social distance, and isolation, quarantine, travel restrictions, and using hand wash or hand hygiene protocols. So during COVID, we are very familiar with this kind of practices. You see, this is the interesting point. So doctors supposed to give some medicine for the disease, but medicine or vaccine is not workable. Therefore, we are compelled to go for the social practices. But apart from this, we have some pharmaceutical measures, vaccination programs. During COVID, Indian government conducted large vaccination programs, and they have given different slots for different people. So we all took the pharmaceutical measure to control the COVID. And in the similar way, we have antiviral therapies, colo- monoclonal antibodies, and even post vaccine also, there are certain precautions we are going to be take, and targeted ring vaccination. So these are the pharmaceutical measures. So naturally, so government or the public health mechanisms or missionary organization, they prescribe, and we are following this. But I would like to present a different concept here, or not different, it is a unique concept and which is less documented in the research. So this I call indigenous idea of herd immunity, and this particular practice is very much available or very much prevalent in the Indian rural areas. So now we look at rural population. Those people that they are not familiar with the social distance, quarantine, or herd immunity, violation, vaccination. Because most of the people, especially the cattle herding communities, they do not have proper literacy. But whenever these smallpox or some sort of pox erupted to their cattle or to the humans, then they go for the quarantine, and they know that quarantine will not protect them from the attack of the virus. And then they inquire about different herbal products. They came to know a unique product which is photographed here, that particular plant called Amatali plant. And they take some leaves and make some sort of juice from the leaves. And they also collect the serum to get the host virus from the animal. So they go to the most infected animal, and they cut somewhere and they collect the serum, and they mix the serum and the leaf juice. And because of this mixture, what happens technically or theoretically, or otherwise in their belief, and the virulence of the virus will be reduced. So then the enfeebled virus they take and they use as the antidote to the virus, and they go to the healthier animal and they make scar on their ears. They infuse this material. When they infuse, they forget about a week to 10 days. Sometimes it is 15 days. After 15 days, they may witness one kind of blister on where they infuse the material. That means on the ears of the animal, some sort of blister will come. So the blister is a cloth indicator for them that is their vaccination is successful. Actually, it is not vaccination, it is violation. The violation is successful. But this is the practice they have been practicing since long time. We don't know when the particular practice was started. But so far, no people, very less people documented this kind of thing. And whenever we look at this practice, so we can compare with the modern science. In the modern science, people are, modern literature is saying vaccine was invented by uh, vaccine was invented. I forgot his name. Okay, so vaccine invented in the medieval in 18th century, during 18th century, then Edward Jenner invented vaccine. So when vaccine, the credit was given to him, and during the invention of the vaccine, Jenner also used animal product to develop the antidote for the humans. That's what there was a controversy whether the paper is going to be accepted or not. However, finally developed, and we are considering father of vaccine is Edward Jenner. So then you look at the Indian society, but this particular practice, the idea of vaccine was very much existing in the from the Indus Valley civilization. So therefore, so we can look at mm, for the indigenous practices. So thereby we are going to be get some sort of new information. So then this information can be cross-checked or cross-verified with the modern medical science. So thereby we are going to be get some sort of insights. So this is another idea. I have gone through field and collected some material. I developed some sort of write up also. But then, so there is a need to integrate both this animal health and environmental health and human health. Of course, overall, we can say it is one health concept. So because most of the diseases are coming from the animals, that's what genetic screening or is very much important. So I feel that so we have to look at this pandemics and epidemics from this point of view. That's what the integration is very much important. Even the project objectives also saying the same thing. But here, one health surveillance system. So one time, for example, we can take the data from the hospitals and veterinary system and wildlife monitoring and environmental thing. These four, if we integrate to the uh, various diseases, definitely we are going to be get a different novel insights, and with that, we are going to be have some sort of new models to confront or to combat the virus eruptions. And so we have we are also know that COVID very much. As even the previous presenter was also saying, COVID originally originated from the bats or some other animal, but however, it is a genetic origin, and that was rapidly spread across the globe, and the world become shut down, and global healthcare become one of the biggest crisis, which was unprecedented and unpredicted, kind of phenomena. But however, we learned some lessons here. One is genomic surveillance is critical. The second one is a need global cooperation. And third one is a transparent data sharing. So these are not happening at the global level. So that's what there is a need to integrate this wildlife data and community awareness programs and even surveillance systems. So that eventually we can come to a different or a new form of conclusion. However, here, what are the takeaways we can take here with the review of literature? One is epidemics are governed by the R or not and the transmission pathways. Of course, because of the mathematical models, and we can know how much serious this pandemic is. So thereby, we can develop some interventions, and we can go with the different schools of thought processes. So thereby, we can develop some strategies. And one of the best strategies achieving the herd immunity, because which is sustained and equitable vaccination will cover. So therefore, we have to think about achieving the herd immunity. In third, to achieve the herd immunity, so One Health is one of the important concept where we can integrate human, animal, and environmental aspects. And with the help of digital technologies and detection and response, and the collaboration and cooperation with the known institutions, and we can navigate into the new kind of strategies that is well preparedness. So this is what I would like to present here. I presented, and so this is just I developed from the review of literature, and we are developing some sort of interview schedules and case study sheets. So thereby we can ask, we can consult the people who are expertise in this area for the indigenous healers, and we can take and we can collect the information from them, and we would like to develop some sort of, what are the this plant-based ingredients they are using, and then we go to the lab, and we have to take the help from the laboratory people whether this is having such kind of properties or not. So that is the second level, step we would like to work on that. So thank you, madam, and thank you one and all for listening.
Thank you, Professor Ram Shepherd.
Yeah, thank you.
Yeah, now it is time to listen to Dr. P. Muralidhar Reddy from Department of Chemistry. And his work is on exploration of anti-diabetic molecules from Indian herbs and species, bioassay screening and sustainable advanced analytical method developments under the theme drug development and sub-theme anti-diabetic drug discovery using photochemicals. I welcome Dr. Muralidhar Reddy to give his talk.
Thank you, madam. Am I audible, madam?
Yes, yes, Murali.
Good afternoon to all. I am happy to present the progress of sanctioned ANRF project. So that it is exploration of anti-diabetic molecules from Indian herbs and spices. So bioassay screening and sustainable advanced analytical method development. So this project is under the theme 7 drug development and under sub-theme 7.2 anti-diabetic drug discovery using phytochemicals. So hub co-PIs are the Dr. Anil Kumar sir and also the Professor Y. Srinivasarao, and spoke PIs main Muralidhar Reddy, Dr. M. Kavita madam, Professor M. Vijulata madam. First of all, I thank the ANRF Hub Institution and also the Spoke Institution of Usman University for sanctioning and supporting this research project. So most of you know that the diabetes is a major health challenge in India and worldwide. Indian herbs and spices are the rich source of diabetic compounds, and many of them are traditionally used
Sir,
for.
Your slides are not changing, sir.
మేడం నేను ఇంకా స్లైడ్స్ ఇంట్రడక్షన్ చెప్తున్నా మేడం. So many of them are traditionally used for the blood sugar management, and through this project we aim scientifically explore these natural resources and screen their anti-diabetic activity and isolate active molecules and develop sustainable analytical method for their detection. So today I will briefly present the So today I will briefly present the background of the work, objectives, and methodology, and green and white QBD based method development, and current progress and expected outcomes and future plan. So this is the outline of my project. So around 830 million adults are affected by the diabetes globally. When it comes to the 77 million people in India are living with the type 2 diabetes, and 25 million people are India are at the risk of or in pre-diabetic condition. So WHO, World Health Organization, identifies that the So diabetes is one of the leading cause for long-term health problems. So it may lead to some heart disease and kidney problems and some eye damage and other infections. So therefore, there is a strong need for safer and effective and affordable diabetic treatments required. So why we need to choose herbals and spice-based discovery? The reason is so Indian herbs are spices are widely used in traditional medicine and a daily diet also. So they contain many bioactive compounds, uh, and therefore the scientific screening can help to identify the active molecule and develop a evidence-based anti-diabetic leads. So, what are the why new active molecules and better analytical techniques are required? The reason is there are the limitations of the allopathic treatment. Also, there are the limitations of the traditional usage. What are those limitations? For example, let us take effective diabetic control possible with modern medicines. However, the long-term use may cause a side effects or some uh, in some kind of patients. So many treatments may not fully address the oxidative stress and other disease-related factors. So and also when it comes to the traditional herbal preparations, we are generally often contains the many of the active and inactive compounds also. So then the exact dose and active molecule and mechanism of actions are not always clear, and therefore there is a research opportunity to identify a specific anti-diabetic bioactive molecules from the Indian herbs and spices, and after that we can do the screen and validate their activity to the proper biological studies, and then you can go for SAR to understand the relationship between the structure and activity. And once you have prepared the analogs, then we can develop a sustainable analytical method for the quality and quantity assessment. And the current project, the main vision is to discover and optimize and monitor the anti-diabetic molecules from the Indian herbs and spices in a cost-effective and sustainable way. So here the project is designed in one is first discovery part, second part is the optimization, and third one is the validation part. So the project has 6 main objectives. First objective number 1 is the review of the literature and select a suitable herbs and spices and collect the sample from the different regions of Telangana and different parts of from different regions from the India. And second, once you have collected the sample, the objective 2 is to prepare the extracts and fractions and screen them for anti-diabetic activity. So then, objective 3 is to perform the chemical fingerprinting, isolation, and structural characterization of the active molecule. And then the objective 4 is once you have done this process to evaluate the isolate compounds for again the anti-diabetic activity, and then develop a structure active relationship studies for understanding. Once if you identified the lead molecules and the lead molecule, then we are going to in the objective 5 we are going to do some kind of chemical modification in the lead molecule, and we are going to synthesize their analogs. And the last, after the synthesis of all these lead molecule, then we are going to develop a sustainable QBD based QBD assisted analytical RPHPLC method for the selective biomolecules. So these are the main 6 objectives. So when it comes to the methodology, so the research work is designed in the 6 phases. In the phase 1, we are going to collect the sample collection and literature survey. And the phase 2, we are going to extract the active compounds and initial screening. And in the phase 3, we are doing some kind of chemical fingerprinting and isolation. Then we are doing the characterization of these active biomolecules. And then in the phase 4, anti-diabetic screening of the isolated compounds. In the phase 5, we are doing the lead optimization and analog synthesis. And the last phase, we are going to develop a sustainable RPHPLC method development. So when it comes to the phase 1, one by one, in the phase 1, literature survey, and we are going to collect the various samples. So recently, according to the review of literature, there are the n number of compounds from the Indian herbs and spices from the Indian herbs and showing the very good anti-diabetic activity. And the major herbs we find is the Petrocarpus marsupium and also Trigonella and Gymnemia. And also when it comes to the spices, pepper, cupepa, and Nigella seeds and cinnamon. So these are the from these herbs and spices. It shows the very good anti-diabetic activity. From this, we have selected. So far, we have collected the few of the herbs and spices. These are the following herbs, and we have collected from the different part of the Telangana region, and one is the Petrocarpus marsupium and Trigonella and Gymnema, and also Tinospora. And also when it comes to the spices, pepper and Nigella and also cinnamon and also curcumum and gingiber. These are the various plants and herbs we have collected from the different parts of the Telangana region. But based on the literature review, suggested that these herbs and spices contain the various bioactive compounds such as alkaloids, flavonoids, and polyphenols, and terpenoids, and also phenolics. So, which may support the glucose regulation through the antioxidant and also the production of a pancreatic beta cells. So, so far we have collected the few of the plant and Indian herbal plants and also the spices from the different parts of the Telangana. This is the phase 1. So in the phase 2, we are doing the bioassay based selection of promising plant samples. So why here in the phase 2, we are going to prepare the various plant extracts and using the solvents of different polarities and screen all the extractions for the anti-diabetic activity, compare the activity and identify the most of the promising samples and select the active extracts and fractions for further chemical analysis. So why this step is important? This step helps to reduce the number of samples for detailed study. It also helps to find which plant extract or fraction has better antibiotic activity, and it also links the biological activity with the selected plant source. So this second phase is one of the most important phase, particularly in this project. So the expected outcome on this phase is the list of active extracts and fractions, and here we can selection of the lead samples for chemical fingerprinting, isolation, and characterization. So in the phase 3, converting these active fractions into identified biomolecules. So here initially we are using various advanced analytical techniques like LCMS and GCMS and NMR and modern chromatographic techniques to study the chemical profile of active extracts and fractions. And once we have done this fingerprinting tool, then we are able to isolate by using the various separation techniques like vacuum LC and preparative HPLC to isolate the active compounds from the promising fractions. So the main aim is to separate and purify the compounds with possible anti-diabetic activity. And once we have done this isolation, then we are going to do the structure confirmation by using various spectroscopic techniques like IR, NMR, and MS based. So the final output will be the chemically modified and validated compound for the biological screening in the phase 3. And the next phase, we are doing the anti-diabetic screening of isolated compounds. Then compare the activity of different compounds. So identify the most promising lead compounds, and then we are going to do, with the help of isolathamide, we are going to do the structure-active relationship, understanding from the screening, and also the here the in the phase 4 the output is the lead compounds with confirmed anti-diabetic activity. Initial SAR studies, understanding for further chemical modification and analog synthesis. This work we are going to do under the phase 4. So in the phase 5, lead optimization and analog synthesis. Once we have identified the anti-diabetic active compound, and then we are going to do the chemical modification of these lead molecules and synthesize new analogs to improve the activity, solubility, and stability. So enhancement of overall pharmaceutical properties. This we are going to do. And here also again, all the synthesized compounds are again characterized and also screened in-house anti-diabetic activity. Compare the activity of the parent lead molecules with the whatever the new analogs which we are synthesized in the phase 5, and results will be interpreted and identify the active pharmacophore. So in this step, the outcome is the optimized lead molecules, SAR supported analog series, and improved compounds for further preclinical and translational studies. So this is about the once after this we are in the phase 6. We are going to do a QBD assisted RPHPLC method for the detection of the selected active biomolecules. See here we are going to develop a optimized RPHPLC method for the active biomolecules. Apply the QBD principles using box Pelican design for the method optimization. Then improve method robustness and also accuracy and cost effectiveness. So use the method for reliable detection and analytical control. So why QBD we are using? The main is the QBD helps in systematic optimization of critical method parameters. So it also improves understanding of the analytical methods. It helps to develop a robust and reliable reproducible method. It supports the better method performance throughout the analytical life cycle. So finally, ultimately, we are going to get it. The end result is a sustainable optimized analytical method, and method is aligned with the modern green and white analytical expectations, and reliable detection method for the selected anti-diabetic molecules. So here we are going to use 3 to 4 green analytical softwares, green matrices, and also the white and analytical matrices. In the green, we are going to use the Agree software and MO modified green analytical procedure index and chlorotalk scale. So these are the three different softwares. The main purpose of this green evaluation method is the eco-friendliness and solvent to evaluate the eco-friendliness, solvent use, so material consumption, chemical safety, and environment impact of the analytical method. When it comes to the white approach or white, this is RGB fast software and also baggy blue applicability grade index software. These softwares to evaluate the analytical performance and sustainability and practical applicability of the method usefulness. So this software at the end of the project, we are going to use this software to the analytical method is exactly what we have developed method is exactly green or this method is applicable for the various other similar kind of analog that we are going to evaluate. So if it is if not green, we are trying to make a develop method. So make it the green. So when it comes overall, our project workflow is the initially we'll select the herbs and spices, prepare the extracts and fractions, screen for the anti-diabetic activity, isolate and characterize the active molecules, optimize the leads and synthesize the analogs and develop a sustainable analytical method. This is the overall workflow of the current Anrf-Pair project. So far, we have collected the few of the Indian herbs and spices from the different regions of the Telangana, and we have started the extraction process, and we have done some. Then this is the workflow filtration, and then so far we have done up to the crude extract part. Then this crude extract we are going to emit for the anti-diabetic activity, and then after the once the results are received, then we are going to separate the active compounds from the crude extracts by using the column chromatography techniques. And then after that, once you have separated this active biomolecule, then we are going to characterize by using NMR and other various spectroscopic techniques. Then go for the again the SAR studies and in vitro screening. Then again the lead molecules. Then we are going to synthesize the analogs from the lead molecules. Then we are going to again perform the anti-diabetic activity and comparison with this. And then finally we are going to develop the sustainable green analytical method for the lead molecules. So these are the time schedule and milestones for the 60 months, 5 years project. So literature survey and sample collection to the publication and report. And these are the 60 months implementation plan of the proposal. Next one is we have the almost the roles and responsibilities of the investigators from the hub and spoke institutes. So myself, I'm from the spoke. So literature study and plant collection and equipment setup and screening of extracts and fraction development of sustainable analytical method development. It's my role. Dr. M. Kavita from also spoke. Kavita madam, and role is the chemical fingerprinting of the extracts, isolation and characterization of the active molecules. Vijayalata madam also, PA madam from the also spoke institute. Madam is going to do the SAR studies of the isolates. So Dr. Anil Kumar Paspu sir from Hub Institute is going to screen the anti-diabetic screening of the isolated compounds. And YSR from the Dr. Y. Srinivasara from the HCU, and he is going to do the chemical modification of the lead compounds and analog synthesis. These are the roles and responsibilities of the all the investigators. So the expected deliverables and outcomes, we have three. One is the scientific deliverables and analytical and translational outputs expected from this project. So when it comes to the scientific deliverables, efficient and cost-effective extraction and isolation methods, and discovery of the novel anti-diabetic molecule from Indian herbs and spices, and isolation of the active analogs and pharmacophore property features and properties. And analytical point of view, we are going to develop a sustainable RPHPLC method for the selective active biomolecules, and we are also going to QBD based method optimization with green and white analytical assessment, and reliable method for the further research and quality. So when it comes to the translational outcomes, so publications in a reputed international journals, and there is a scope for the IPR and patent generation or patent filing, and also potential progression towards the preclinical and clinical studies. These are the expected deliverables and outcomes of this project. And when it comes to the potential impact and use of the research outcomes, how the project can create the value beyond the laboratory. One is the health impact and the scientific impact. So, and also innovation impact and academic impact and also societal impact. So when it comes to the health impact, so potential to generate the safer and specific and affordable anti-diabetic candidates. When it comes to the scientific impact, strengthens the natural product-based drug discovery with modern analytical validation methods. And in the innovation part, supports for the IP generation, patent filing, and translates research and lead optimization. So in the academic point of view, enables the publications and conference presentations, and also long-term, also we can develop a long-term collaborative research with the hub and other institutions. And when it comes to the societal impact, promotes the scientific use of the Indian herbs and spices for addressing the major public health problem like diabetes. These are the potential impact and use of the research outcomes. So these are two publications we have. I have not exactly based on this project, but the project money we have. But these are under the same theme. It's an anti-cancer activity related. The two publications there we have acknowledged the pair project also, but the some part of the pair money also we have utilized uh, to get these publications. These are the two publications, and the conclusion. What the key takeaways from this project is the NRF address the major diabetes burden with Indian relevant and photochemical based strategy, and it's a the project follows a well planned and 6-phase approach from sample selection to the validated analytical method development, and supports discovery of the new molecule, new analogs, and future IP and publications, and builds a pipeline that can ultimately support the clinical and societal impact. So about the conclusion of the project, so I also acknowledge University of Hyderabad and Usmania University, and Senior Professor Brahmananda Manavati sir, Project Director Anrf-Pair, and also Senior Professor M. Vijayalata Madam, PA Anrf-Pair Project, and Dr. Anil Kumar sir, and also the Professor Y. Srinivasarao, University of Hyderabad. So thank you one and all for the opportunity.
Thank you, Dr. Murali. Now it is time to listen to Kavita, Dr. M. Kavita. Her topic is, one second, deciphering gymnic acid, natural remedy for diabetes from Gymnema sylvester. And her theme is drug development, and sub-theme is anti-diabetic drug discovery. using photochemicals. I invite Dr. Kavita to deliver a talk. Dr. Kavita.
Thank you, ma'am, for nice introduction. Good evening, good afternoon, all. Welcome you all to the webinar presentation as a part of the Prajapalana Pragati Pranalika by Government of Telangana. I am going to talk about our collaborative Anrf-Pair project titled Deciphering Gymnamic Acid, a natural remedy for diabetes from Gymnemia sylvestri. Our collaborators include from the Abu University, University of Hyderabad, Dr. Anil Kumar Paspuleti, Department of Biochemistry, and Professor Srinivasarao Aragorla from Department of Chemistry, University of Hyderabad, and from the Spoke University, myself, and Professor Vijulata Madam, and Dr. P. Muralidhar Reddy from the Department of Chemistry. And my topic comes under theme 7, that is drug development, and sub-theme developing antidiabetic molecules from phytochemicals, and task number 55. Madam, why I am not able to move my slides? So we already know India is the diabetes capital of the world. Nearly 90 million adults are affected with a prevalence of 10.5%. And most of these adults are aged 20 to 79 years. And around 43% of the cases were still undiagnosed, and which is going to cost a huge health expenditure around 9.8 billion. This is the data from year 2024. And this is going to reach around 15.2 billion by 2050. And beyond these numbers, we can also have a major health impact. Like mostly this diabetes is a major contributor to cardiovascular disease, kidney failure, and also neuropathy. So we know that it's not a simple or just a medical condition, but it is a national health crisis, demanding affordable, innovative, and sustainable solutions. When we talk about the current diabetic medications which are present in the market, the first line of treatment is mainly with metformin. This is the first line oral antibiotic drug. Its mechanism is through improved insulin sensitivity, but the main side effects are going to be, it is going to have nausea, diarrhea, and also they have discovered vitamin B12 deficiency. And even this metformin is not suitable for all the patients if the patients are having any renal or hepatic impairment. And also there is a relevance like long-term tolerance issues also reported with the metformin. Along with metformin, when we go with other drugs which are there presented in the market, like sulfonyl ureas and thiazolidine diones and SGL2 inhibitors and GLP-1 agonists, when we look at these drugs are effective, but they are very high cost, and also they are associated with hypoglycemia. That means reduced sugar levels so much. And also they are going to have some kind of cardiovascular risks. And because of these side effects of the drugs which are available in the market, And as the diabetes is rising in India, it also demands like low cost or accessible therapies. And we already know that traditional knowledge systems, especially in Ayurveda, it is going to offer a rich source of bioactive compounds with centuries of safe use. So among those, to introduce the integrating the traditional knowledge which already there in Ayurveda and people are using it into modern therapeutics is our idea to develop a proper diabetic medicine. For this project, I have specifically selected Gymnema sylvestri plant, and which in Hindi it is commonly known as Gudmar, and in English it is saying as sugar destroyer. And it's already validated in modern science. And this plant, it belongs to a family Apocynaceae, and which is a native to tropical forests of India. And mostly its active components which are working against diabetes are gymnamic acids. These are triterpenoid saponins. And these gymnamic acids are also very famous for suppressing sweet taste perception. That means if we eat these we feel like a lower sugar cravings, that we don't feel like eating sugar. And that's why it is traditionally used in Ayurveda for Madhumeha. As you know, diabetes is known as Madhumeha in our Ayurveda. And modern studies on these plant, on these compounds, gymnamic acids, they are also show anti-diabetic activity. Along with the anti-diabetic activity, they are also showing hypolipidemic activity and anti-obesity properties also this plant is showing. That's why I have selected this plant for further project. These are some of the gymnamic acid or gymnema products which are available on the market. You can see that as a herbal medicine and herbal tea and also Ayurvedic medicine, you can see different type of products which are available in the market, like gymnema tea and homeopathic medicine. So our idea is to combine these effects with the traditional systems. I have checked whether we can we replace gymnema with metformin. Then most of the said no, metformin you have to use, and this gymnema may help in reducing the dosage of the metformin. And when we look at various species of gymnema, we have Gymnema sylvestri, Gymnema montanum, Gymnema latifolium, and Gymnema unnanis. Among all these species, when we look, only Gymnema sylvestri is very well-known, studied, and human trials also has substantial clinical evidence also is there for Gymnema sylvestri. Whereas other species, when we look montanum, latifolium, and unnanis, they also show promising preclinical anti-diabetic activity, but most of the human trials are not carried on these plants. And Gymnema unnanis, it's mainly occurs in China. Here also, they only started early stage screening of the phytochemicals. But we need to have this, and future research, our future research should focus on standardization and also comparative phytochemistry and validation across all the species. When we come to the mechanism of action of this gymnamic acid or gymnema plants, mostly the mechanism is it is going to reduce the intestinal inhibit the intestinal glucose absorption, so that the post-meal glucose levels are going to reduce by using this. And also we already discussed that it is going to suppress the sweet taste, so sugar cravings can be reduced. And also this plant is increasing the stimulates the insulin secretion. So endogenous insulin can be increased and also helpful in beta cell regeneration or protection, so that the beta cell mass is going to increase, and peripheral glucose uptake is going to increase insulin sensitivity. Along with all these mechanisms, this is also acting as a antioxidant and anti-inflammatory. The main active components or key components for all these things is mostly gymnamic acids, and antioxidant and anti-inflammatory property is mainly due to flavonoids and stigmasterol, which is present in the extracts, and it is also going to, because of its antioxidant activity, it is also going to protect the pancreas. So gymnema, it acts through multi-target mechanisms like blocking sugar absorption, stimulating insulin, regenerating beta cells, and improving insulin sensitivity. But main thing is large scale clinical trials and standardized formulations are still needed before it can be fully integrated into mainstream diabetes therapy. These are the various structures of gymnamic acid. Mainly the core includes gymnamazinin core. It's a tritapinide backbone. You can see many hydroxyl groups on the backbone. These hydroxyl groups can be replaced with various kinds of sugars like glucose, galactose, arabinose, so that the core structure can give us different types of gymnamic acids, like around 20 types of gymnamic acids have been identified. And if we vary the sugar moieties on the core, it is going to vary determine its biological activity also. So you can see the core structure again, gymnamazinin, and gymnamazinin has more of hydroxyl groups. So hydroxyl groups mainly, they are useful for hydrogen bonding, and the hydrophobic backbone which is present, this is mostly for the transporter binding. And this tritapinide skeleton of gymnamagnesin is the structural foundation for gymnamic acids. And it's a kind of, you can see 5 cyclic rings are there. So it's a pentacyclic olenin framework, which is having mostly hydroxyl or carboxyl functional groups, which can be allowed to attach the sugars, more sugars, and creating lots of varieties of gymnamic acids which are responsible for the antibiotic diabetic activity. There are at least 20%, 20 plus gymnamic acids are identified with different structural moieties. Structural differences are there. That is mostly on the sugar structures. And again, the biological activity is going to vary depending on sugar moiety composition and the affinity. So our specific objectives for the project is first to extract and characterize gymnamic acid and its analogs from various gymnema species, and also mainly concentrating on Gymnema sylvestre. Once the extracts are prepared, then we need to prepare anti-diabetic assays to study, and also structure activity relationship studies. By using AI/ML tools, we can change the core structure. We can change the side substituents on the main gymnamazinin core, and we can see how the structure is going to affect the function, and also study binding studies between understand the bindings between the gymnamic acid, different varieties of gymnamic acids with various like beta cell receptors and all. And also optimize the lead compounds through chemical modification and study again using AI/ML tools, and develop analogs for potential clinical evaluation. Methodology includes collection of Gymnema sylvestre plant, and then using extraction and isolation and screening of gymnamic acid compounds, then chromatographic fingerprinting of the compounds. Again, once the purified compounds, again do the second antidiabetic assay and identify the lead compounds and analog synthesis. Once the lead compounds are identified, their analogs can be synthesized. Methodology includes literature survey and collection of selective plant, and Gymnema sylvestre extraction, identification, and screening. We already have seen the gymnamic acid, it is highly polar. So the first step is we have to, dry the leaves and prepare the extracts. Already Muralidhar Reddy sir has discussed the extraction procedure, and then extraction is going to be carried out using polar solvents, and these again extracts are examined for presence of gymnamic acid. We can use some few tests like froth test and acidity tests. After preliminary confirmation, these extracts are further fractioned for yielding the specific compounds, and the fractionation of the compounds is confirmed by performing TLC. We all the chromatographic fingerprinting of the active compounds, it can be done using high-end chromatographic techniques and spectroscopic instruments, and the compounds are further characterized by using Fourier transform infrared spectroscopy and NMR spectroscopy. And the isolated compounds are taken for anti-diabetic assay, and the selected compounds are proceeded with the SAR studies. And computational chemistry and structural analysis can be done using AI/ML methods and also neural networks to predict the binding of gymnamic acid to various proteins which are involved in diabetes. The chemical modification of lead compounds and analog synthesis. This study is going to be focused on lead optimization, and all the synthesized derivatives will be subjected to in-house biological screening. And by analyzing the results, active pharmacore will be identified. Chromatographic fingerprinting of the compounds is going to be done by me, and I'm going to take help of Professor Vijayalata madam for the SAR studies, and also using AI/ML methods and neural networks. And chemical modification and analog synthesis will be done by Professor Y. Srinivas, and anti-diabetic assays will be done by Professor Anil Kumar Pasupuleti. This is the time period which is approximately given for 5 years. During the first year, we are going to mostly do literature survey and purchasing the chemicals and the collection of the plants. And till now, I have collected the plant and prepared the crude extracts. We need to go for the anti-diabetic assays. Professor Anil Kumar sir is going to carry out the anti-diabetic assays. When we compare the comparative analysis, current drugs versus Gymnema, mechanism of action, when we compare, the metformin is going to target single, like it is going to target only hepatic glucose, whereas Gymnema, it can increase, it can decrease, it can going for a multi-target, like it will decrease absorption and increase insulin. Mainly Gymnema sugars, they mimic glucose. That's why they are going to inhibit the glucose absorption. And I have already discussed about the side effects of the traditional drugs, the current marketed drugs. And when we go with the traditional systems, minimal side effects are reported, but it needs a validation. In cost-wise, if we look, anti-diabetic drugs automatically, especially if you go for sulfonylureas, like GLP-1 agonists and SGL2 inhibitors, they are of high cost. Whereas these are low cost and because plant-based and potentially affordable. And accessibility of the drugs in the rural is limited, whereas widely available in Indian traditional medicine systems. But only problem here is evidence for the current anti-diabetic drugs. We have large scale clinical trials are there and they have regulatory approvals. Whereas for Gymnema, we have very small human trials and we require a strong preclinical evidence and these are not yet standardized. And when we go for long-term use, definitely we have issues with tolerance and adherence for the current anti-diabetic drugs. Whereas for Gymnema, we know that from centuries people are using, but the main problem here is the dosage, how much dosage, it's not clear, like dosage is unclear. Main research gaps and challenges when we look, standardization is the main issue because the gymnemic acid content is going to vary depending on species, region, and also extraction procedure. And also till now doses and safety protocols are not established and long-term safety data is also limited. Bioavailability, it's poor absorption and we don't have about the stability of saponins may reduce the efficacy. And the mechanistic clarity also, multi-target actions are promising, but we need more molecular confirmation. Like when we go with the beta cell enhancing pathways, it's not very clear. We need to study that also using these things. And computational predictions, obviously it needs experimental validation to ensure the translational systems. We need to address these research gaps and challenges. For this, we are going to have some future directions because the protocols are also not properly established. So we have to develop reproducible extraction and purification protocols for gymnemic acids and initiate a large-scale randomized control studies to validate efficacy and safety in human populations. And using computational models, we can predict binding affinities and optimize analogs, and which can lead to accessible to lead discovery, and explore Gymnema-based dietary supplements. If these things are, then we can go with nutraceuticals, like where we can use these Gymnema-based dietary supplements for affordable diabetes management and also for low income communities, and investigate the synergetic effects of Gymnema with existing antibiotic drugs to reduce the chemical dosage and also side effects. Thank you so much. I thank Professor Vijulata ma'am and Anil Kumar Paspuleti and Aragorla Srinivas and Dr. P. Muralidhar Reddy and all the co-investigators for giving me this opportunity. Thank you.
Thank you, Dr. Kavita. This finishes today's webinar. Tomorrow again at 10 o'clock we'll meet so that the rest of the co-PIs can present their talks. Thank you very much.
Thank you, ma'am.